Okay, so we left off with cell size. We left off with cell size, and we said, what's the limiting factor as far as how large cells can be? Why can't they be larger than they are? Go ahead.
Because when they get to a certain point, then they split into two different.
Yeah, that's what happens. They do actually divide when they get too large, but why can't they just be larger? Why couldn't we just be one giant cell?
Because they're usually.
What do we have to be able to do? Go.
They have to go across the surface.
Yep. We have to get nutrients in. We have to get waste out. And if the cell gets too large, there's not enough surface area to get nutrients into that entire volume and out and waste out of that entire volume. All right. So types of cells, and for some of you, this might be, for the people that are in the other section, I think this might be review, but prokaryotic cells. Pro means before seed. And although the nucleus isn't a seed, you maybe can understand how people looked at it as though it were the seed of the cell. It'd be kind of like the pit of a peach or something like that. This dark staining part that's in the middle. So prokaryotic cells did not have a nucleus and do not have membrane-bound organelles. Now we might hypothesize that these were the first cells. Why do you suppose it would be reasonable to think that these are the first cells? Olivia? Because there's no nucleus. So what would you say about them kind of more generally? You're right. How would they compare in terms of complexity to other cells? These cells are simpler, right? It seems to be reasonable, doesn't it, that organisms would develop simple before complex? So in terms of the development of living things, we in fact are the pinnacle of creation, kind of the last, and because of the higher level of complexity. So prokaryotic cells include archaea and bacteria. And then the kinds of cells that we have are eukaryotic cells, cells that do have a nucleus and membrane-bound organelles. And those are the ones we're going to spend probably more time talking about and more time looking at are eukaryotic cells. When we're in the lab on Wednesday, we'll look at eukaryotic cells. We'll look at bacteria cells. And bacterial cells are also organisms. The cell is the organism. So you call those unicellular organisms. The cell is the organism. So they don't have the levels of organization, cell, tissue, organ system, organism like we do. The cell is the organism. Go ahead, Sam.
So how are we going to see the cells?
Or the microscope. Is that what you mean?
So you're going to zoom up, put your hand out, and you zoom in, and you see a bunch of cells?
Well, we do have actually a live specimen microscope that you can see the surface of your epithelial tissue, but you can't really see the individual cells of it. So we may look at that too, but you're going to be looking at a slide. You'll take a scraping of the inside of your mouth, not like the video had the other day where the narrator was telling about the person looked at the gunk in their teeth. We're not doing that. We're just looking at the epithelial cells that line your mouth. And they wouldn't be that much different than the epithelial cells that line the surface of your skin, but you can scrape a toothpick against the inside of your mouth and collect some of that and then look at those cells. So we'll look at those, and then we'll look at some cells of an onion. And so then you can kind of get a comparison of what plant cells look like versus what animal cells look like. Okay, so prokaryotic cells. Prokaryotic cells eukaryotic cells would include bacteria. They're small, but they are visible with a light microscope, which is what you're going to be using. The plasma membrane of a bacteria is surrounded by a cell wall. It's not like a cell wall like you would see in plants, but it is a cell wall nonetheless. It'd be worth your while to get your book out and take a look at page 62. Page 62 shows what a bacterial cell looks like. The one on the left is what's called a non-photosynthetic bacterium, and it's got these large tails called flagellum on it. And then the one on the right is cyanobacteria, which is sometimes called blue-green algae. So you can see it as a cell wall, but it won't look like what you'll see in the lab on Wednesday. The plant cell walls that you see in the lab on Wednesday will be much more kind of angular, rectangular particularly. These are kind of rounded, but it's a little sturdier covering. And you might imagine that the reason for that is, it helps to protect them. This is the whole organism, remember, that we're talking about. And it talks about them being able to attach to surfaces using fimbria. And that's only true of some of them. Bacteria have fimbria, which are short appendages that help them to attach to an appropriate surface. Now, the reason for that could be something like, if these are bacteria that live in the digestive tract of an animal, if they didn't have some way to hook on, what would happen? They go right through. That's right, Caleb. They have to be able to stay in the digestive tract, and so they have this ability to anchor. And that's how digestive system parasites work also, or intestinal parasites work that way also. They have hooks like a tapeworm has these kind of hooks on its head to keep it in the digestive tract.
I'm going to show you one. I got a picture of one. Oh, yeah.
Of what's that?
See, tapeworm that came out of the cow.
Yeah, let's.
Motile bacteria.
Can we see it, Mr. Put on the other one. Let's put on the other one, Mr. Nye. We can all see it.
She works at
I want to see it. Do I want to see it?
Right there's the one.
Where is it?
That big white
Oh,
thing.
laying on top of the gate there. Okay.
Just trying to put it on the
Boy,
projector.
that's a real dandy.
Put on the other one.
I can't see it.
You can't see that very well, but anyway, it's on top of a gate. Caleb's showing us a tapeworm that came out of a cow.
Let me see, too.
But anyway, the reason that's fitting is we're talking about things that live in the digestive tract being able to anchor themselves. Okay, so the DNA, guys, you need to be looking at page 62. 62. You don't have your book out, get your book out. Page 62. There is no nucleus. There is no nucleus, but there is still genetic material. There is still a chromosome. What does nucleoid region mean? What does it mean when we say nucleoid? Maybe. That's not the answer. Nucleoid means like a nucleus. It's not a nucleus, so it's not enclosed. But if you look at those two pictures on page 62, guys, can you have your book out? You're looking at the pictures? That's not true. They're not on my face. Okay. So you can see that there's kind of a dense area where the DNA is found in each case, but it's not enclosed. So although they have a nucleoid region, they don't have an enclosed nucleus. So they could also have additional DNA rings called plasmids. Okay? Now, the next thing this is still prokaryotic cells. The cytoplasm is filled with ribosomes. And just like in eukaryotic cells, ribosomes are where the proteins are made. And that's really the main function of cells, is to make proteins, to make stuff that you need. Photosynthetic bacteria have thylakoids, and it shows that on page 62 in the picture on the right. The green parts there are thylakoids. We'll talk more about that when we get to photosynthesis, but that's a thylakoid. And a thylakoid is a stack of kind of discs where photosynthesis occurs. They're full of chlorophyll, which is the pigment that plants have that allows them to photosynthesize. We have pigments in our skin. We have melanin, which is what makes our skin darker. We also have keratin, which can give your skin kind of an orangish hue. Any of you ever seen a baby that's eaten a whole jar of baby food carrots? They turn orange. None of you have ever seen that?
Not commercial.
No, I don't think.
Well, it's true. They get a lot of keratin. What's that? Well, yeah, because I mean their body's not very big, so eating a lot of keratin can actually cause their skin to kind of turn color. Yes. Yeah. Yeah. The president might have too much keratin. That's right. Yes.
How much do I feed you to turn orange?
You probably have to eat a lot of carrots.
Oh man, it'd be like a whole field of 2008.
Okay. So the cytoplasm. Now, here's something that's kind of interesting. The cytoplasm in bacteria, they're real protein factories. Now, although they're simpler and can't do maybe as complex of things as our eukaryotic cells can do, they can make proteins pretty rapidly. So prokaryotes have adapted to living in almost any kind of environment. So they're very simple, though they've been able to adapt and develop systems that are resistant to extreme pHs, extreme temperatures, okay, all of that kind of thing. All right, we're ready to move on to eukaryotic cells, which is what we have. So make sure that I would put a line in your notes or something, make this really large letters, the eukaryotic cell, so that we can distinguish between without nuclei and now with a nuclei. And we're going to talk about these in terms of three areas that exist in a cell. And so any ideas what the areas of a eukaryotic cell would be? None at all? Okay, well, the areas of a eukaryotic cell are the outer boundary, which is the phospholipid plasma membrane, which we should know from last chapter. Show me with your hands what that looks like. The phospholipid bilayer, right? Like this. Your fists represent what part of the phospholipid? The phosphate. And your fingers represent the what kind of tails? What's the other thing that makes up a fat? Fatty acid tails, that's right.
Yeah.
What's that? Oh, this is just a heading. There's really nothing there. But it says regions of. That helps you any. Who are we missing from this section? Carson and who?
Haley, Cash.
What are you taking
Cash
the
isn't
video?
running today, is he?
No, he claims he's sick. What are you taking the video for?
For the kids that are gone. So Cash is sick, and Carson's running, and Kaylee.
Kaylee's
Is that what you said? Okay. Very good.
And we're working. Yeah.
and we're doing our thing.
I mean, we should all get candy. Mr. Brown, we should all get candy because we're working instead of the kids who sit at home.
He's not actually sick.
We should get candy. Do you have some?
No, you have some.
Oh, it's my turn again. Oh.
When
Why
hasn't
you're
it
the
been your
teacher?
turn?
You're the boss, right? You need to give us all
Thank
candy.
you. I appreciate that.
You need
I
to
don't
give
always
us
hear that.
That's a candy.
Yeah.
I should give you candy,
You should.
but I'm not going.
So am I. Why not?
Yeah, Sam don't need no more candy.
I should give you candy because you were really doing a good job pulling that.
So I should
I like that.
Mr. Mallett, were you saying like Sam Scholes is just a beast on the radio?
I don't know if I put it in quite that verbiage.
Why
I
not?
didn't
Mr.
realize
Mallett?
it was you.
Next
When we
time,
said Bill
next time
O'Malley,
Sam
I was
just
like,
totally whiffs.
wait,
She said you
Mr.
did a good
O'Malley,
job.
you were talking?
Yeah, it's always
I
Mr.
thought it
O'Malley.
was you.
Mr. O'Malley and Tanner.
What? Tanner's
Them
really
thing-
good.
them-
That's how we do it.
Them two
Mr.
go back
O'Malley,
and forth.
that's straight disrespect right there.
And then he'll go by you and slap you on the back.
Do you give audience candy when you teach them?
Yeah, Mr. O'Malley just goes out the window. Just
Okay, we're moving on. What's the primary material that's in a plant cell wall?
Cellulose.
Cellulose. Now, there's also a secondary wall that can be made of lignin, but the main thing you have to remember is cellulose. Okay, now, what would we consider kind of the guts of the cell? This is between the boundary. Yeah, the insides, the guts, the viscera. No, that's not really it, but it's the cytoplasm. So the cytoplasm is the semi-fluid medium. What do we mean when we say medium? We don't mean like they do on TV, the people that supposedly talk to spirits.
The medium?
They call themselves mediums,
Really?
but
Yeah.
that's
Like
not
the
what we're
middle.
talking about.
Like the middle.
What we really mean is something. So you might say that if you were making chicken noodle soup, the medium would be the broth. That's what you put all the other stuff in,
I
right?
see.
So the medium is the thing that's holding the other stuff. So the medium in this case is what's called cytosol. Cytosol is the medium of cytoplasm, and then it has all the organelles in it, all of the endoplasmic reticulum and ribosomes and all of the parts that make up a cell. Okay, and then the nucleus, which is the control center. That's what's really controlling things. Okay, so metabolic functioning. What do we mean by that? The metabolic function of a cell. What? What's the number one metabolic function that's going on in our cells that's releasing energy from our cells? We'll spend a whole chapter talking about it.
But I guess-
So your cells need oxygen and nutrients, and then the waste, they produce nitrogen containing waste and carbon dioxide as a result of what's called cellular respiration. So the big thing with regard to metabolism is cellular respiration. That's how we get energy out. But that metabolic function could also be in plants photosynthesis, which is controlled also by the nucleus. Okay, so what's in the nucleus? Well, we have chromatin, which is the thread-like material that contains DNA and protein. And when it's ready to divide, that chromatin can kind of coil up and form these rod-like structures called chromosomes. And the reason for that is, well, so this is kind of what chromatin would look like. just a sort of a random arrangement of this strand of DNA. Well, when it gets ready to divide, what it's going to do is it's going to look more like this. It's going to become more of a kind of a rod of material. It's still the same. It's still the same DNA. It's still the same amount of DNA. It's still the same sequence of DNA. But the reason for that is when the cell divides, guys, it's important that we each get a complete copy, that each new cell gets a complete copy. And when it's in the chromatin form, if we were trying to divide it, it would be like if you had your textbook there and you had all the pages, but they were just all scattered around. And so I'm gonna give a copy of the textbook to Carly and to Caleb. And I know I have 600 total pages, which is the amount that it takes to make 2 books, but they're just all interspersed. Well, it's going to be pretty hard to make sure that I give Carly 300 unique pages and Caleb 300 unique pages. It would be better if we had the book already bound into pages 1 through 300 and 1 through 300, and then we can easily separate them. So that's what we have when we look at a chromosome. We have this nice packaged piece of DNA. Whereas when it was in the chromatin state, when it's all scattered like this, and we have other amounts of this, the same thing, it would be hard to ensure that we're getting the right message to both new cells. Does anyone know why, when the cell's just going about its normal business, why it would be better for the DNA to be uncoiled like this? Any idea why it would need to be uncoiled? Well, what's the purpose of the DNA? What's it coding for? It's telling the cell how to make which type of macromolecules is it directing the cell to make? Well, let's go back a chapter. What are the 4 macromolecules? Go.
Proteins, lipids, carbohydrates.
And nucleic acids, right? Which one? This is nucleic acid, right?
Yeah.
Deoxyribonucleic acid. Well, what's it coding for? Which of those things is this telling the cell how to make? Well, think of the functions. What's the function of carbohydrates?
Ready energy.
Ready energy. How about lipids? Stored energy. How about proteins? Hold. Building something, right? So this is telling the cell how to make proteins. Well, let's say that it needs to know how to make a cardiac muscle cell. Well, if it's all coiled up like this, that message on how to make a cardiac muscle cell might be buried somewhere clear in there. How do we get the machinery of the cell in there to copy that to tell the cell to make that? So if it unwinds, now this same strand that was buried inside is now exposed, and this strand can be read by the cell and knows how to make that particular protein that's necessary to make a cardiac muscle cell, which, by the way, your body doesn't do a very good job of. That's why when somebody has heart damage, it doesn't recover very well. The message would be there, but the mechanism to turn that on. Does anybody know what other kind of cells aren't very easily repaired in your body?
Brain cells?
Brain cells, nervous tissue, right? That's why. Does anybody ever had an injury, maybe a cut or something, where they've lost feeling in a digit or something like that? Or do you know anybody who doesn't have a feeling in one of their digits, one of their fingers? Fairly common if you have any kind of a cut. What was they like on Mr. Deeds when he had the black foot? Probably never saw that. Okay. Anyway, so that's chromatin and chromosomes. And when they, in an individual, it's important that they have the same type and amount of DNA. So nucleoplasm is the same stuff in the nucleus that, so I'll give you an analogy. Nucleoplasm is to nucleus as blank is to cell. Nucleoplasm is to nucleus as blank is to cell. Look back through your notes. It's something you just wrote down. And look at the definition. Look at the definition. Nucleoplasm is to nucleus as blank is to cell. Tanya? Cytoplasm. It's the semi-fluid goop in the nucleus that cytoplasm is in the cell in general. All right. Now, this is probably something you're going to be able to see. This is something you'll be able to see probably under a microscope when we look at cells. And you should be on page 50. Flip your book to page 50 to look at the picture. So this is the picture you should be looking at, guys, in your book.
Wait, just one more. What is the
Say that again?
nucleoplasm? It's a semi-fluid medium
Of the nucleus. Yeah. Yeah, that part was cut off until I. Okay. So the nucleolus is. So if we did this analogy, we could say that nucleus is to cell as nucleolus is to nucleus. So this is that kind of the nucleus is that kind of a seed part, the kind of a control center of the cell, the dark staining part of the cell, and then within the nucleus, the darker staining part of it is the nucleolus, where ribosomes are made. So I would, on this, okay, so I would circle that because that's something you're going to have to know, of course, the chapter. Okay? And then the fact that the nuclear membrane is a double membrane, what's that like? Ellie? What would the nuclear envelope be kind of like if it's a double membrane? Well, let's go back to this. Is this a double membrane?
Yeah.
What is that? What are we showing? The phospholipid bilayer, the cell membrane. So again, with another analogy, the nuclear envelope is to the nucleus as the cell membrane is to the cell. Go.
What does it say
After what?
passage?
Oh, I'm sorry. I didn't know my face was in the way there. I'll go back to this. I just wanted to be able to circle that. What the heck? As pores that allow the passage of proteins and ribosomal subunits. So stuff has to get in and out of the nucleus, just like stuff has to get in and out of the cell. Because where's the message? Where's the message for what a cell needs to do? Where is that found? Zarek, where are the brains of the cell, so to speak? They're in the nucleus. So if you're going to get the instructions out to the cytoplasm, which is where stuff's actually made, there have to be these pores. Does everyone see that? You should be looking on page 50, guys. Does everyone see the nuclear pore? Carly, can you see that? You will be as soon as you can get your book out.
You're going too fast. Can't write that fast.
We're rolling.
What page?
Because we work
50.
so hard, maybe everybody should be.
That would be probably good. On page 50, the nucleus, The four parts it shows about the nucleus are nuclear pore, that's where stuff gets in and out. The chromatin, that's the message, that's the DNA. The nucleolus, what happens there? Megan? You look in your notes if you want. What happens to the nucleolus? Should have it circled. Ribosomes are made. So that's where the things are made that make proteins. Ribosomes make proteins, but the ribosomes themselves are made in the nucleolus. And then the nuclear envelope, which is a double membrane, just like the cell membrane. Okay, so now we're going to get into some of the organelles in particular. Ribosomes, and they're just going to look like little tiny dots. Those ribosomes get instructions from the nucleus. They make proteins. They're small. They're made of two what are called subunits. They can be scattered in the cytoplasm, or they can be in groups called polyribosomes. And they can also be found on what's called the endoplasmic reticulum. I'm going to have you write, and then I need you to find all three of those things. Show me where there are ribosomes on the endoplasmic reticulum, where there are ribosomes just in the cytoplasm, and where there's a polyribosome. So the three places are So those are the three spots. So the three places where there are ribosomes are scattered in the cytoplasm in groups called polyribosomes and also on membranes. It's called rough ER. So get your book up on your desk and show me those three things. You have to have three fingers on the diagram. The one in the upper left shows where there are polyribosomes scattered in the cytoplasm, where there are ones on the membrane, and where there's a polyribosome, which means a chain of them. Show me those three things. Anybody got all three? Three fingers. You have to turn around and look at Kirsten's. You got to bring your book. Show me on the diagram, not where it says it is, but where it actually is. What do you have there? Okay, so the little dots on there, that's them. And dots all by themselves. You have all three, Kirsten? Don't show me the name. So look here. There's a chain. Here's where they're dotted on the membrane. Here's where they're all by themselves. Okay? All keep looking at this. So here's where they're by themselves. Here's where they're in a chain. Here's where they're on the membrane. Okay, there's where they're in a chain on the membrane and by themselves. Yep. Okay, very good. Chain, where are they on the membrane? There, by themselves. Scattered by themselves. By themselves. And chain on the membrane.
I don't even know.
Three things in yellow. Here's where they're in a chain. Polyribosome. Here's where they're on a membrane. That's rough ER. They're scattered by themselves. Well, that's by themselves over here. Anywhere they're scattered. These little dots scattered around. Okay.
It's time to go.
No, it's not. Okay. The last thing. It's too early. Okay. Don't hold your breath. Okay. For tomorrow.
We need that for every single day this week.
Mr. O'Malley, if someone selected this meeting. Do we have a leaf project?
Part of the reason you have a lot of it to do is because you didn't do it in the last month.
Wait.
Yeah, I was working on mine this morning. My dog.
Not just the leaf project.
Actually, his dog did like run
Yeah,
down.
put it.
Mr. O'Malley, I'm not even kidding you. His dog broke.
Why do we have to have such a big project for like one page chapter?
So no, that's actually a good point.
My dog.
Mr. O'Malley, I have no trees in my house.
Okay. You should answer on page 1, 1 to 15. Page 1, 1 to 15.
That's not real clear.
You have one? You got it.
Mr. O'Malley, we're so busy. Oh, that's true.